Maybe there. Hi, Jade. How are you doing?
I'm good. How are you?
All right. You staying warm?
About as warm as I can get.
It's to the point where it's kind of like the house only gets so warm, right?
Yes, unfortunately. And I have one whole wall that's windows, so all the cold air just seeps in, and I'm like, please no, stay out.
Yeah, we live in an old house. It's 90 years old, I guess, and 80, 90 somewhere in there, and it's just drafty everywhere. Yeah. I always wanted to when I was a kid. I always wanted to live in a castle, and everybody said, "Oh, they're drafty," and I said, "Well, I can put up a little draft." Yeah, right. I don't have the castle, but I've got the drafts, so
You're halfway
but
there.
you can feel the breeze, you know. All right. I think we're gonna have. Well, nope. Yeah, everybody. No, we're still missing a person. Yeah, the cell wall actually is not a solid barrier. We think of it as that. I like to use that box and balloon analogy. I think I talked to you about, yeah, a week ago, whenever it was we had meeting last. But the plant cell wall actually has some gaps in it. Okay, and these gaps allow um, for tiny strands of the cytoplasm. Hey, I've got a misspelling there that I can fix because of the mode I'm in. So we'll just do that. To extend between the cells. And this allows for something called cytoplasmic streaming that we'll talk more about later. Okay, I believe when we get on into that. There'll be a portion of the chapter that talks about that. That allows for, and this I think this chapter goes into a little bit more explanation of that too, but it allows for nutrients to go back and forth between cells. And this is something that we don't see that in animal cell. I mean, we only have the membrane. That membrane is semi-permeable, but things have to get through that membrane. Okay, this is open. I mean, this is allowing for stuff to just flow back and forth between the cells. So it's kind of cool and neat. And yeah, there's a cell membrane in there that can kind of slow that down a little bit, but yeah, we'll just see more about that when we get to it. Okay, it's a little bit of it's definitely a unique thing. Okay, with plants. Um, we also have something called dictosomes that we don't have in animals. Uh, Again, with these are some of those many types of vesicles. I say we've got the vesicles, the vacuoles, and the saccules, and this and that. Well, so these are flattened discs that are scattered throughout the cytoplasm, and they have different functions. And I think again we're probably going to get into those in detail further on. So right now they're like introducing the terms to you. Okay. All right, and then our last slide in this group here is where we're going to talk about all these plastids. There's all sorts of different types of things called plasts, okay, which the chloroplast is the best known of them. So these are organelles that are associated primarily with the storage or manufacture of carbohydrates of some sort. Of course, chloroplast makes sugar, glucose, major carbohydrate there. It contains chlorophyll that does that. You've heard about chloroplast before, so and you'll hear about it again. Okay, so we're just going to go over these real quick. The chromoplast. Anytime you see the term chromo, it usually has something to do with color. Excuse me. Okay, and so this one, there are other colors, okay, but this synthesizes and accumulates the carotenoid pigments, and the carotenoid pigments are the pigments that give carrots their color, the orange color. Okay. Leucoplasts, we have two types. We have amyloplasts that synthesize starches, and elioplasts that synthesize oils. Okay. So plants don't synthesize the fats, like the animal fats. Okay, they're going to do the oils. So we have elioplasts in there to do that. Okay. Um, Another interesting thing about these plastids is they have the ability to change from one type to another under certain conditions. And again, I think we will probably get into that. Um, I've mentioned that the chloroplast has its own DNA. Okay. So these are kind of all derivatives off of each other. And they all develop from something called a proplastid. Okay, so you kind of got this vague basic plastid that can become pretty much anything. Okay. Um, and so whatever's needed. So again, I think the mechanisms of that is going to come in a little bit later, but I believe how your book is set up with all of this is that um, they'll start just using this vocabulary, assuming you know what it is at this point, basically, and then tell you how it's functioning. You're like, well, what the heck is that? I know what it does now, but what is it? So this is your basic introduction, and then it'll come back and build on it in a future chapter. Okay, so let's going to finish off that presentation, and let's get on into. Yeah, I want to save those changes. Into our tissues. Okay, so major groups of plant organs. We have roots, stems, leaves, and flowers. And we're going to spend the next few weeks going over these things, the roots, the stems, the leaves, and the flowers. And the lab I had planned for you yesterday, I had hoped we would actually do it on campus in person. I was going to assign you all your individual microscopes. We were going to find a place to, you know, keep those locked up, which I think probably in my standard classroom is probably the best place, just back up in there. But we put tape on it so you know which one's yours. Those have been locked up since before COVID, so they probably don't need to be disinfected when we get them out. But you can always disinfect it after you use or not. You know, it's like I said, it'll be dedicated to you. Nobody else is using those all semester. And I had slides of different types of tissues. Okay, for you to look at. Actually, these are all stem tissues. Okay, we're going to do stem tissues first. That's what we're going to do. So, but that may be next week. If we'd had class yesterday and they hadn't called it off, I would have sent out a message to you saying that we were going to meet in Zoom. And actually, what I had designed for yesterday's lab was just a real quick little find a window and observe what plants you can out of it. Okay. Um, with a little bit more instruction than that. But I don't know. We may wind up using that one again if we don't have if we have bad weather. That may be what next week is coming will come to be. So we'll just have to see how that goes. But kind of be prepared for that. It's not you don't have to go out. You can if you want. I don't limit you to your window, but my recommendation is stay warm and find a nice window and look out. And mostly I was going to ask you look for trees and shrubs because that's pretty much what you see right now. Depending on where you are, you might have some tall grasses that are still, visible from last fall, but mostly just shrubs and trees. Okay, which are all tree categories actually. Okay, so anyway, that's the lab tie-in there. But so in talking about stems, okay, the first or kind of the plant overall, but it's the stem. The first thing I want you to know is meristems. Plants have permanent regions of growth. Well, plants do have this. I don't know what that is in there. Plants have permanent regions of growth where cells actively divide. Um, so a tree, you may or may not know this, but a tree does not grow from the ground up. Okay, it doesn't push itself up from the bottom of the trunk. It gains mass with tree rings and growth every year there, but it grows from the top. Okay, it gets taller at the top. Okay, roots also do the same thing. You know, they don't grow from the base and push on out. Down at the root tip, right behind there's a protective coating on the end of that tip, but right behind that is where your cells are rapidly dividing, and that's where the elongation of the root occurs. Okay, so it's the ends, and so when you're looking at like the top of the tree or the ends of the branches, okay, those are meristems. That's where your growth is actively occurring. So we have a tree will have something called an apical meristem, and the root will too, okay, on the branches and the roots. So these are found at or near the tips of roots and shoots. And they produce new cells, which increases length and primary growth. And in trees, you usually have one apical meristem that's considered like a primary meristem. And that's where the most growth is going to occur. And that's if you're looking at a tree that's kind of got that triangle shape to it, or even the rounded tree, still that center point is taller than the rest, right? Um, that main tallest stem, that's your apical meristem. The other ones are referred to as lateral meristems, and they don't have as rapid an elongation and cell division as the apical one will. And that is also one way of taking a plant that would normally be a tree and making a shrub or bush out of it, is that while it's young, you cut that apical meristem, and that will cause a kind of a stunting of the growth of the tree. And it'll also cause it to round out, okay, instead of keeping that triangular, angular shape. Um, so that's if you have a tree that actually gets that apical meristem destroyed out of it in a storm, then you can also see sometimes that stunting or rounding happens. It doesn't always, because there's hormones involved here, okay, enzymes and things that may trigger a nearby lateral meristem to take over and become the apical. So it also depends on your species of your tree, whether or not you can actually make a shrub out of it, or whether it'll just resist and start a new growth somewhere else. Okay. Um, so that's kind of the meristems. All right, but that's your primary growth area. We have three types of primary meristems. We have what's called the protoderm, the ground meristem, and the procambium. Yeah, here's the lateral meristems. They produce tissues that increase the girth of roots and stems, secondary growth, um, and two types of lateral meristems are vascular cambium and court cambium. Okay. And then our grasses are herbaceous type of plants that aren't, well, actually, this is more monocots, okay? Um, and sort of maybe some herbaceous dicots, but this is most of your monocots. Your grasses are monocots, okay? Um, Instead of having lateral meristems, because these are often just annuals, they grow up there once a year, then they die back. You're not going to get that increase in girth. So what you're looking at here is what's called intercalary meristems, okay, and instead of lateral meristems. Okay, so again, probably more on that when we get specifically to that grouping of plants. Okay, so in these different types of meristems that we've got, we have tissues. So the meristems are considered like suborgans, I guess. Okay, your organ is your stem. Then what do you have? And then you have at the tip end, you have the meristem. What kind of tissues make up the meristem? Um, we have the parenchyma, and the cells that make that up are called parenchyma cells. Okay, so let's review this here. The basic unit of an organism is a cell, but if you have a group of cells working together for a specific function, okay, then they can make up a tissue. And you have a group of cells, actually it's usually maybe two different types of cells, but it can also be just one type of cell work together to make up a tissue. And then different tissues working together will make up an organ. And then your different organs make an organ system. So your hierarchy of organization. Okay, recall that. I think we had that reviewed in chapter 1. And so your parenchyma cells are actually the most abundant cell type. They function mostly in food and water storage and make up the edible parts of most fruits and vegetables. So when you're eating fruit and eating vegetables, you're mostly eating the parenchyma cells. And to think about that, it makes sense because we're wanting to eat the part that has a lot of starch or sugar in it because that's nutrition for us. And then oftentimes there's a lot of water in there too. Fruits are, if we like them, usually they're pretty juicy. Vegetables, same way. We want that water. It's a source of water for our body too. Um, some of these will contain chloroplasts and function in photosynthesis, and those have a special name called chlorenchyma. Okay. And parenchyma cells can be pretty long-lived. Okay. Cells usually have a lifespan. Uh, and I think I mentioned that last year or last semester in biology class. In genetics, we'll probably do it again. I'll talk about it again this semester in genetics if I get there. But um, because we're really behind now, and who knows what the weather's going to continue doing. So I may gloss over some of that stuff and not do it again. But your DNA, every time it copies, you lose a little bit of a telomere off. That sound familiar? The telomeres, and so when that when all the telomeres are gone, then the cell dies, and any cell, well, the cell will live out its life, but when it goes to replicate and make a new cell, make two new daughter cells, those two cells can't function because something other than a telomere is lost. Something that actually codes for something is lost when it's copied. So that doesn't work too well. And so the longer a cell can live before it has to divide, then the longer that organism can live. And so they found that there are some parenchyma cells in cacti that can live to be over 100 years before they have to divide. So that means plants can live a really long time, barring disease, drought, um, all sorts of things like that. There are some plants that are pretty much almost immortal, okay, or have the potential to be immortal. Um, but they don't. There's a bristlecone pine out in New Mexico, the mountains of New Mexico, that they've been able to pretty certainly date one at least to 4,500 years old. That's older than the giant redwoods, which live pretty long as well. They suspect some of those trees may be older. They just don't want to go around core sampling all of them because that could shorten their life. Okay, so they kind of wait till one naturally dies, and then they count the tree rings and look at it. So yeah, if we'll probably get into immortality of plants a little bit later. Okay. And so if you ever take a cutting of plant, like, the vines that you can take and you clip it off and then you stick it in some water and it'll make new roots. That's the parenchyma cells that are doing that. Okay. They begin to divide and make new roots. Now they've got to have a hormone present that stimulates that. Some plants naturally have that. And other plants don't naturally have that, and that's why you have to use the rooting hormone. So again, in your after dinner gardening project that you're doing, you might want to investigate that about your plant. Is yours one that will make roots on its own from these parenchyma cells because it naturally has the hormone to do that, or will you need to provide the hormone for that? Okay, this is for the bee plant, the vegetatively propagated plant. Parenchyma is another group here. These often occur just beneath the epidermis, which, epidermis on us is the skin. Well, epidermis on the plant is its outer coating of cells. This provides flexible support for organs such as leaves and the floral parts. So the best example that you probably can easily picture and identify with is the stringy part of the celery plant. Okay, that you get caught in your teeth when you eat celery. That's your parenchyma cells. Okay. You can see how that would provide support, right? And then we have sclerenchyma. This consists of cells. I know they all sound alike here, and how am I ever going to keep these straight and separate? It's challenging. I know. I remember. Okay. Sclerenchyma consists of cells with walls made thick by lignin. Lignin is a waxy coating, okay, on the cells. And these are definitely support cells. And by the time they reach maturity, they're dead. Okay, they're dead and they're coated with this stuff. But that allows them to be strong and provide support. There's two types. We have what we call a sclerenchyma. We have those that we call sclerids. And where you will find them is if you eat pears and you notice the grit in the pears. Okay, that's a grouping of these sclerenchyma cells. And they're called sclerids. The shells of nuts are made up of this type of cell, and peach pits are made up of this type of cell, the sclerids. And then we also have sclerenchyma that make up fibers, and these are like plant fibers that are used for textiles. So if you are familiar with linen fabric, that's made from the flax plant. And so the stem of the flax has these long strings, and it's like, well, it kind of like, it's not, but it can stringy kind of like your celery, but that's different cells. Okay, different cell type. But these fibers can be extracted out of the stem, pulled out of the stem, and then woven into a fabric with proper processing and treating. Cotton, okay, your cotton in the cotton ball, cotton boll, um, is another one. Hemp, obviously, too, has the fibers like flax does. Okay, those are your those are our main plant-based fibers. You can use other things. Native peoples all over the world have found all sorts of plant fibers that can be used for stuff. Okay, for making textiles. Um, Gosh, in this area, thousands of years ago, I found evidence that the plant called we call rattlesnake master now um, is a big, tall, kind of fibrous plant. It's more common out west now than it is here because it was a little hotter and drier here a few thousand years ago. Um, but we have found that the cave dwellers over in Arkansas area and some here in the northeast Oklahoma, where there's some of the caves around the Arkansas River area, they made fishing nets, okay, out of fibers extracted out of the plant called rattlesnake master. Okay, so yeah, that type of thing. Um, I've actually seen one of those nets preserved over in the museum at University of Arkansas. I was there. I went for an ethnobotany conference there because botany, ethnobotany. I'm into ethnobotany. That's that. When I graduate with my PhD, it's going to become definitely more of a hobby of mine. Um, anyway, they had it out. We could put gloves on and touch it, but it's like this thing is thousands of years old, and I know that every time it's touched is and I wasn't a professional sitting there doing research, which some of the people I was with were. So I didn't touch it. And I don't regret that really, but it's kind of like I could have. It was just cool to watch it because it was still flexible. I mean, it had been so well preserved in the cave that they found it in that it could, I mean, theoretically still be used today. Um, so and they were keeping it well preserved there. So it was just amazing to me to see that and to see how well constructed it was. Um, so complex tissues, these are composed of two or more kinds of cells. And so xylem, really big tissue, of course, in a plant. So there's our xylem and our phloem. The xylem is the chief conducting tissue for water and minerals that's absorbed by the roots. Okay. So with xylem, it's basically everything's taken at the roots and then brought up through the plant where it's needed. We have 5 cell types in xylem, and I'm only going to have you study 3 of them. Okay, I want you to know about vessels, tracheids, and ray cells. The vessels are made up of vessel elements, and these are just basically long tubes for water transport. Okay, kind of like a straw. Um, and then you can have these tracheids, which are kind of like in a lot of plants, those are kind of like on a support, kind of like support cells to the vessel elements. But in your cone-bearing plants and some non-flowering plants, your tracheids are going to actually make up the xylem instead of the vessel elements. The tracheids have closed ends instead of being open tubes. Okay, they have well, somewhat closed ends. It's like if you took your straw and you stuck it in some, let's say, wax, but then you took a pen and you stuck a few holes into the bottom of that wax. Okay, or the wax. That's more what the tracheids are like. They've got little holes or pores to allow transport through, but the transport's gonna be slower. Okay, it kind of slows down the flow, basically. Um, and I think probably when we get into those plants, we'll talk more about why that's an important feature for those types of plants. Okay, but it has to do with cone-bearing plants typically being evergreen, often living in very dry environments and things like that. Just kind of slows down the flow, and that's good and bad at the same time. Okay, ray cells are again things that provide for the lateral transport. So if xylem is going to go pretty much straight up the veins. Okay, and veins can go out to the sides, obviously, but that's your xylem. But so if like you're looking at a leaf, you've got the leaf. I have my drawing pad up here, so I'm just gonna switch to it since I can. I had it up here for doing DNA and RNA stuff. You remember that? Okay, for last hour. Okay. And you're not seeing that, are you? Okay, let me see what I can do then. Okay, are you seeing a white screen now? Yeah, okay, great. Okay, so gotta close that off. Can't see what I'm doing there. Okay. All right. Now I've got to be artistic on a screen that I'm not calibrated on yet, right? Yeah, okay. All right. So, yeah, let's erase that. I don't like that. And do. Oh, this is gonna be a lot harder than I thought because I haven't practiced drawing on this. This will work. Okay, so here's my leaf, all right, my leaf stem, and down here from my stem point, I'm gonna have, and get that lined up, I'm gonna have this vein coming out, going, all the way to the tip, right? Okay, so my xylem is gonna be in that, my xylem and phloem both are gonna be in that main mid vein. Okay, we can get some branching veins coming off of that. Depending on what type of leaf it is, it could be alternate or opposite. I'm drawing opposite veins. Okay, well, that almost turned out alternate. Okay, and they go out so far. Okay, so water can be transported directly here, and that means, by osmosis, we can get water into the nearby cells, but that's not going to really feed the whole leaf. So we've got to have other cells taking care of that. Okay, and that's going to be your ray cells. All right, so sometimes off of these, you'll see some leaves may just have that main vein, and the rest of it is ray cells. But here, sometimes you'll look and you'll see like little netty looking things coming off of these veins, these side veins. That's um, that's where these ray cells come in. So again, when we get into the leaf, studying the leaf in detail, we'll probably get into a lot more of that, and probably stem too. We'll see. Okay. All right. So let me get rid of that and put you back onto the other screen. Okay. All right. Am I back to my complex tissues slide? Yes.
Yep.
Great. Thanks. Okay. All right. So then the phloem, which I was talking about in conjunction to the xylem, this is, sorry, hit my mic there. This is going to kind of almost go the opposite direction. So we have photosynthesis occurring up in the leaves mostly. Actually, it can occur anywhere you've got green cells that have chloroplasts in them, but the leaves are the main area of gathering light for photosynthesis. And so all the sugar and stuff that's produced in that isn't needed immediately in the cell that's produced in is going to be sent to the phloem, and that's going to flow down. Okay, phloem flows down. It's kind of, you can keep the two apart, and be stored. And if you're talking about a tree, then that extra stuff gets stored in the roots. Extra sugar gets stored in the roots for the winter. So when it is dormant in the winter right now, that's where all that is. And then when the sap starts coming up in the spring, then that's going to come back up to fuel the growth of new leaves and all of that. So yeah, phloem can flow about anyway. any direction it needs. But we usually think about, okay, it's all the stuff is produced up here and it's all taken down for storage. It's the direction we think about it going. But also it can go lateral. Okay, if it's produced in this cell, but this cell over here is not producing enough or much, then it can transport sideways immediately. Um, so just because we think of it, usually the greatest part of it going down to the roots, that's not where all of it goes, and it's not the only way it can travel. It definitely travels around however it's needed. So it conducts the photosynthetic products throughout the plant. And we have two types of cells there involved. We have sieve tube members and companion cells. And the sieve tube members are placed end to end like the vessel elements. Okay. Um, and the interesting thing about the sieve tube members is that they don't contain nuclei. Okay, every cell has a nucleus, right? These don't. It's really kind of strange. So plants are pretty complex, all right? They're just, yeah. Um, they have cells adjacent to them, partner cells there, called companion cells, and that's where you find the nucleus. And that nucleus works to direct both cells. Okay. Um, it directs the activities of the sieve tube members. Gosh, I got a lot of typos. I typed this fast, you can tell. Just didn't go back and double check it. Um, and controls themselves. So anyway, that's another one of those, I don't know, neat, weird, strange things about plants. There's a lot. Plants are just really different. The epidermis is the outer tissue of a plant. Usually it's 1 cell thick. Okay. You can think of it as the plant skin if you want. Okay. Then we have periderm, which replaces epiderm in woody plants. And that's what's going to constitute the outer bark. And it's primarily composed of cork cells. And that does get a little bit thicker than 1 cell. Okay, well, and again, when we get into those, we'll get more detail onto that. Then we have secretory cells and tissues for secretion. These function to isolate or move from the plant substances that are produced by the plant. So these would be like we're talking about secretion, we talk about hormones. So these are things that one tissue may produce a certain type of hormone that's needed in another place of the plant or several places in the plant, not necessarily the site of production. And so those things have to be transported somehow, and they have to be moved. Okay, so these secretory cells are what's going to move those substances around or help move them around. And some of these plant secretions have value to humans. Okay, so we'll again talk more about those when we get to those particular plants. So eventually we're going to get through all these tissue parts, and then we're going to start going into the plant groupings. So if you were in the biology class in the fall, then we start off with we have non-vascular and we have vasculars. And the non-vascular, that's where we'll start probably is the non-vasculars. Though we'll probably go through a little algae before we get into that. All right, so anyway, we'll go into a lot more detail when we start talking about these non-vasculars. Last time I said non-vasculars, we've got 3 divisions or phyla, but botanists like to call them divisions. And we had the Anthosaurophyta, Hepatophyta, and Bryophyta. Remember those? And that was it. I said liverworts, hornworts, and true mosses, and we're done. We'll talk a little bit more about specifics on those in this, as you can probably see if you look at in your book. So that's where we'll be going into the details of those. Okay, and that's all I have for you today. Do you have questions?
No, I don't
No.
think so.
Okay. Did I have a quiz up for you all this week for today?
No.
Yeah, I didn't think I did. I was gonna write one and put one up, and then when the weather just started doing weird stuff, I was like, yeah, I don't know that we're gonna cover enough to do a quiz. So um, we won't. Hey, tell me something else about these labs you did last week. Were they the same lab? I'm sorry, was number one was part one and pond water. Were those like the same labs for you? Was that a repeat of looking at pond water twice?
I don't think they were for me, at least.
Okay, they're not
No, I don't
supposed to
think
be.
so.
You don't think so? Okay, good. Somehow or another for my biology class, I put them up and they're the same lab. They're having to do the same lab. And I'm like, okay, it's not supposed to be because the operation of the bright field microscope is supposed to take you through the microscope parts and introduce you to the microscope. So I don't know if I like mislabeled the same lab, and then yeah, I have to go in and sort it out. But I just want to make sure that McGraw-Hill didn't do something where they changed the lab on me and I didn't know. But if it was different, there were different labs for you, then that's how it's supposed to be. So just checking. Okay. All right. Well, everybody stay warm and safe over the weekend, and I'll see you Monday.
Have a nice weekend.
You too. Okay, Lexi, are you there?
Yes.
Okay, so I did grades because I had to report people who had low grades, and I noticed that you're missing a bit of stuff from. Oh, let me see. Let me get my grade book down here. Last week. You didn't do labs last week, and you didn't do the quiz on the Friday before that. Were you gone? Were you ill? Was something going on there?
Last week I had a lot of stuff going on, and I guess I just missed it. Like I didn't see it or something. Because I tried to get it all done, but I guess I just didn't see it.
Okay. Um, because that's just really here within the past week. And it looks like the week before, no, the second week of school, you also missed like 2 quizzes, but that's kind of getting a little far back. But these right here, I can open those back up for you. If
Okay.
you think can you get them done like this weekend? Do you think?
Yeah, I can get them done this weekend.
Okay. Hey, yeah, and if you do miss stuff like that, I mean, this is small class, okay? A larger class, I'm kind of like, I don't have time to go back and do stuff for people individually like that. But with a small group of people, it's a lot easier to manipulate things around like in the software and stuff. So,
Yeah.
um, yeah, you just let me know in the future.
All right.
As soon as that happens, you can say, hey, I just overlooked that. It closed out on me and I didn't know. And I'll probably work with you to open it back up. Okay. All
All
right, so
right.
I'll
Thank
make a
you.
note to get that done this afternoon. I'm not sure really what time that'll be available for you, but I'll
All
definitely
right.
get done this afternoon, so it'll be available for you over the weekend. Okay.
Okay. Thank you.
All right. Have any questions of anything?
No, I do not.
Okay. Well, that was my only question for you is kind of what happened and did you want it open back up?
All right. Thank
All
you.
right. No problem. Take care.
You too. Stay warm.
Thanks. You too. Bye.